Welcome back. So what we've been doing in the past few videos is we've been getting to know the biosynthetic routes for some of the neurotransmitters and hormones, and then also we've been looking at their degradation pathways. Now, if you've looked at the drug detoxification videos, I want to have you think about something, and even if you haven't seen it, it'll be good for you to hear it anyways. Let's think about the ways that you prepare a molecule for excretion. Well, there's several ways. Number one, P450 enzymes can perform hydroxylation reactions. So hydroxylation is one way to increase the polarity of a molecule. Because if you take a carbon chain that's already hydrophobic and you attach an OH group to it, that makes it more polar and more likely for the kidneys to be able to excrete it. So that's one method. Another way is if you have a sulfotransferase, they can sulfate through the process of sulfation. Okay, so they can sulfate molecules, and if you actually look at what a sulfate looks like, we did this in a previous video, but you can basically see, even without me drawing it, you can see the vast majority of all these dipole moments. In fact, let me go ahead and draw those anyways. You have these dipole moments that point away from the sulfur and towards the oxygen atoms, and there's basically four of them, and if you count this R group over here, then there's another one. Okay, so it's very polar. You have all sorts of partial charges. In the case of most sulfates at physiological pH, you also have a full negative charge. So they're very polar, very charged, and they allow things to be excreted. Another example of something that you could do would be glucuronidation. So glucuronidation. And glucuronidation uses a slightly different technique, although it's for the same purpose. You use a molecule called UDP glucuronide, and UDP glucuronide transfers a glucuronsyl residue onto the molecule, and that increases its polarity drastically. And so that also aids in increasing its solubility and enabling the kidneys to excrete it. So these are the three main ways that you'll generally see for most drugs and even some neurotransmitters. But there's also another way that's a lot less known, and that's called phosphoribosyl transfer. So this is phosphoribosyl transfer. You generally don't see phosphoribosyl transfer until you get to nucleic acid biosynthesis, because that's normally where it's used. And phosphoribosyl transfer, let me go ahead and show you what it would look like. If you have a phosphoribosyl group, it looks like this. So this right here, so here is part of the characteristic ribose ring, and then you'll have a phosphate right here on the 5 position. Okay, so if you actually look at this right here, this carbon is number 1, this is 2, this is 3, this is 4, and this is 5. So this is a ribose 5-phosphate, but when you use a phosphoribosyl transferase, it starts off with basically this pyrophosphate linkage over here on the 1 position. Okay, so this molecule right here that I'm drawing, this is called PRPP, or phosphoribosyl pyrophosphate, PRPP. And PRPP is basically the universal ribose donor. Okay, so if you want to transfer a ribose onto something, what you're actually transferring is this component right here, and it comes from PRPP. What we're going to find is that one of the routes for histamine degradation actually uses this functional group in order to increase the polarity of the molecule. Just for a moment, let's think about that. So let me redraw the ribose ring, and I'm actually going to make an effort to draw out all the hydrogens. Okay, so if we're looking at this, let's make sure to draw all the atoms. And now let's think about this molecule in terms of its polarity. And just keep in mind there's some R group over here. It's going to end up being the imidazole, okay, in this pathway. Well, let's think about all the dipole moments. Well, here I have a dipole moment pointing towards that oxygen. I also have another one pointing towards the oxygen from the proton. And the same argument can be made for this hydroxyl group as well. So if I look at the partial charges on those, I'll do those in... Let me do those in green. So I'll have a pretty big partial positive on these protons, partial negative on these oxygens, even a partial positive on these carbons right here. And you could go and label all the partial charges. You could do that, for a while. You would see this has a partial positive, this has a partial negative, this has a pretty big partial positive. But the whole point is that the ribose 5-phosphate ring is incredibly polar. And so that's going to help excrete the imidazole ring from histamine. Because what we'll find is that when you make histamine, it's an irreversible decarboxylation. Because if you remember from the previous video, we took histamine. And actually, let me go ahead and draw the structure just so you can get accustomed to seeing it. So what I'm doing right now is I'm drawing the alpha carbon backbone for histamine. So what you have is something like this. So here, so right there, this is histamine. Okay, this is histamine. So if you use histamine decarboxylase, so let me write that. This is histidine. This right here, this is L-histidine. So L-histidine decarboxylase. Keep in mind these are pyridoxal phosphate dependent decarboxylations. We're going to lose carbon dioxide. Specifically, you're losing the alpha carboxyl group as carbon dioxide. And so what you should get is this. So you'll still have the alpha amine, but you'll get something like this. So you'll get that. And this molecule right here, this is histamine. And that's what we saw in the previous video. We saw basically the reaction where you produce histamine. Okay? It's just one step, really easy. And histamine is used as a neurotransmitter. It promotes wakefulness. In fact, when you block a histamine receptor, it actually promotes drowsiness. And that's actually the part of the mechanism of why most antihistamines produce drowsiness. Because histamine is a neurotransmitter in the brain that promotes wakefulness. So when you block its receptor, histamine can't exert its effect, and therefore you get drowsy. In fact, one of the interesting things about histamine, anyone that's ever taken a benzodiazepine, which is a GABA agonist, or at least a GABA allosteric activator, let's just put it that way. Anyone who's ever taken a benzodiazepine versus an antihistamine, even though they both produce drowsiness, you can actually physically tell the difference in feeling between the GABA allosteric activator, which is the benzodiazepine, and the antihistamine. It's a very bizarre thing, but you can actually tell the difference physically. Antihistamines tend to be stronger than GABA allosteric agonists like benzodiazepines. Of course, it's going to depend in large part on the identity of the benzodiazepine, but if you take something like Xanax, an antihistamine is certainly much stronger than Xanax. And Xanax is a pretty typical benzodiazepine to use for things like anxiety. Some people even use it to help them relax to sleep. So it's certainly not a sleeping pill, but histamine or antihistamines will knock you out a lot more than Xanax will. Okay, and something like histamine also it tends to promote inflammation, and in fact that's its main function, is for use in the immune system. So when people take antihistamine, they're looking to suppress inflammation, but the fact that it blocks the histamine receptor in the brain is just a byproduct of the fact that it's blocking those particular type of histamine receptor blockers are blocking all of them. So not only are they blocking the inflammation histamine receptors, but they also block the ones that cause wakefulness in the brain. Okay, so that's just a little bit of the review of histamine. You can go in a lot of detail about that. The last video was pretty short. But we know how histamine is made. It's made by histidine decarboxylase. But it turns out that there's no way that you can reverse it. You can't just put the carboxyl group back. You've got to deal with what you have. And what you have is histamine, so you've got to get rid of that somehow. And it turns out that there's side-by-side catabolic pathways that you can do. Now, one thing I want to say just for the purpose of the mechanisms here is typically the way I draw histamine or histidine or whatever is from the perspective of a tautomer. Okay, so there's two tautomers that exist in histamine. Okay, what you can essentially do is something like this. I'll draw the electron movements in yellow. You could have some base over here that deprotonates here, and we're just doing a simple base catalyzed tautomerization. That's going to force these electrons in here, and these come out and abstract a proton from another base. or in that case, it's acting as an acid. And so what you get is this form of histamine right here. You could do the same thing for histidine. It wouldn't matter. But the point is that you can do this for imidazole rings. And it turns out that this is the form of histamine that I want to use if I'm expected to do this pathway. Okay? So what I've done is I kind of simplified this, and I put the pathways side by side as they relate to each other. Okay? So the pathway that's going to go right next to histamine is going to be starting with a molecule called N-methylhistamine. So this is N-methylhistamine. Okay? And you notice the methyl group, I'll do this in pink, this is the methyl group right here that's been attached to this nitrogen. Okay? And it's an S-adenosylmethionine dependent methyl transfer. So S-adenosylmethionine is the methyl donor, and we of course spit out S-adenosyl homocysteine. That gets hydrolyzed into the SAM cycle, and you regenerate SAM. We know the story from there. Now, when you do this, you get N-methylhistamine, and now what's going to happen is you're going to react these two molecules, both histamine, which is right here, and N-methylhistamine. You're going to react those with some enzymes that are basically enzymes referred to as alkylamine oxidases. So let me actually come over here and talk a little bit about alkylamine oxidase. In fact, let me do this. So these are alkylamine, oops, didn't mean to do that, alkylamine oxidases. And this is the basis of an alkylamine oxidase. So you could have some R group. It's usually a carbon chain. And then what you'll have is an amine attached to it. Okay? And the reason they call it alkylamine is because, of course, there's an amine right here, and then on the other side of it, there's an alkyl chain of some sort. Okay? So the enzymes that oxidize these are alkylamine oxidases. And through the mechanism of these enzymes, what tends to happen is they'll form a Schiff base. Okay? So what'll happen is you'll end up getting something like this. So this right here is a Schiff base, and if you're not familiar with Schiff bases, essentially what they are is they're double bonds between a nitrogen and a carbon. So right here, this is the carbon atom I'm talking about, and this is the nitrogen. There's a double bond between them. That's a Schiff base. This nitrogen has a positive charge because it has two protons. Therefore, we call it a protonated Schiff base. Protonated Schiff bases, which is what they'll be as in the active site of the enzyme, they're really prone to Schiff base hydrolysis. So what ends up happening is when you get a Schiff base like this that's protonated, water is going to be allowed into the active site, and what you'll essentially get is a Schiff base hydrolysis. So what ends up happening is this Schiff base is essentially replaced with an aldehyde like this. And if you wanted, you could even draw the hydrogen in right there, but it's not necessary. You get an aldehyde, and then you end up getting this amine, which is hydrolyzed off in the form of ammonia. So in general, for the neurotransmitter amine oxidases, for the alkylamine oxidases that react with the neurotransmitters and hormones, these are your two general products. You're going to get some aldehyde and ammonia. That's usually what you get. There's only one exception I can think of to this, which is epinephrine. You'll get methylamine, but you'll just do another cycle of this and get another aldehyde and ammonia. So they all follow the same general mechanism. Okay? And the substrates and reagents and products and so forth for these reactions I've shown right here. So what you do for these reactions is you put in oxygen and water, and you get out ammonia and hydrogen peroxide. Okay, the water that's shown right here is the same water that's used to perform this Schiff base hydrolysis, and the ammonia that comes off there, that's the ammonia that's right here. Okay, the oxygen that's used is not used directly on the histamine or the N-methylhistamine. The oxygen is essentially used to oxidize the flavoprotein that's used, or whatever coenzyme is used, and you end up spitting out hydrogen peroxide. Okay, so the oxygen is just used to regenerate the resting state of the enzyme, and the hydrogen peroxide is a byproduct. And by the way, the hydrogen peroxide ends up getting metabolized by catalase, and you end up with oxygen and water. So that's something important to know. The enzyme that metabolizes hydrogen peroxide is catalase. But that's not of importance here. What we're worried about is neurotransmitter and hormone metabolism. Now, one of the things I want to point your attention to is both histamine right here that's shown here, both histamine and N-methylhistamine, they can both react with an enzyme called histaminase. And histaminase is simply one of these alkylamine oxidases. Okay? So histaminase uses a special coenzyme called topoquinone, which is very similar in nature to FAD. It has the same type of chemistry, even similar to pyridoxal phosphate. But histaminase is one of the alkylamine oxidases that will metabolize histamine and N-methylhistamine. The distinction between these two molecules, though, is that monoamine oxidase B reacts very strongly with N-methylhistamine. The methyl group here plays a big role in determining whether or not the molecule will react with monoamine oxidase B. But the net reaction, and I want to point your attention to this, remember what we just showed is that alkylamine oxidases first oxidize to a Schiff base and replace the amine with an aldehyde. So what you should expect in these cases is this group right here should effectively be replaced with an aldehyde. So when I scroll down, I should expect to see those, and in fact I do. So these are the aldehydes that I was talking about. Okay, these are the aldehydes. So these are both, the one on the left is the metabolite of histamine, the product of histaminase. This one on the right is the product of histaminase and monoamine oxidase B when it metabolizes N-methylhistamine. And note that the methyl group is still present right here. Now, both of these molecules can react with the same enzymes. And they'll react with an aldehyde dehydrogenase. And just like in most cases, when you have an aldehyde dehydrogenase, so let's say I'll have some R group here, and I have an aldehyde. In general, what aldehyde dehydrogenases do, so I'm going to put aldehyde dehydrogenase. In general, what they do is in most cases they use NAD+ as the oxidizing agent and water, and you end up getting out NADH. In some cases they use NADP and you get out NADPH, but in most cases it's NAD, and the functional group that you end up obtaining at the end is a carboxylate. Okay, so what aldehyde dehydrogenases do is they oxidize aldehydes into carboxylates. Okay, that's something really important to keep in mind. That's what we see here, is that you take this aldehyde, whether it's the metabolite of histamine or it's the metabolite of N-methylhistamine. In both cases, the aldehyde, and I'll do this in purple, in both cases the aldehyde gets oxidized into a carboxylate. So note that structure right here. Okay, and the other thing that's important about this particular step is it can also be catalyzed by a rather strange enzyme. You wouldn't expect to see it here, but it's called xanthine oxidase, and it reacts with both of these metabolites. Xanthine oxidase is an enzyme that's involved in purine catabolism, and I don't really see the similarity here, but it turns out that xanthine oxidase does react with these particular metabolites, and it will still give you the carboxylate here that's shown in purple. But note that the reagents that you use for this type of reaction are fundamentally different than what you would use in the aldehyde dehydrogenase. So that's why I didn't include net reagents here, is because both of these guys use different reagents. One of them is an oxidase, so it's going to use molecular oxygen. Okay, so having said that, now what we have is this molecule on the left here. This molecule has a special name. It's a major metabolite of histamine, and this one on the right is called imidazole acetate, and that's one word, so I'll put the dash there. Imidazole acetate. And this one over here, you could probably guess its name. It's N-methyl imidazole acetate. So that's the only distinction here, is that the methyl group is present, but you name them the exact same way. And the reason it's imidazole acetate is if you were to break this molecule down, let me do this in light blue. The imidazole ring is right here. Okay, I think if you're in biochemistry at this point, I think you should be able to recognize that that is an imidazole ring. In gray, I'll draw the acetate. So this is the acetate group. Okay, this is the acetate group. Okay, so you combine those two, you get imidazole acetate. Put a methyl group there, it's N-methyl imidazole acetate. Okay, this product right here is capable of being excreted. So it'll get excreted by the kidneys. It's capable of doing that. It's polar enough to where that can happen. Okay? But in the other case where there's no methyl group, there's actually one step further that the cell takes in order to ensure its excretion. And that's by reacting it with an enzyme, which I didn't write here. This is called, first of all, I'm going to designate, this is IA, imidazole acetate. So this is going to be imidazole acetate phosphoribosyltransferase. And that's what I wanted to show you down here. For phosphoribosyltransferases, they use this guy, which is phosphoribosyl pyrophosphate, PRPP, and it transfers this group in yellow that's surrounded by yellow. It transfers this ribose 5-phosphate group onto things. Okay, so this is a phosphoribosyltransferase, and so you end up getting this guy with a phosphate and a ribose ring. Now it's incredibly soluble, and this too, I'll box it in yellow, this too can also be excreted by the kidneys. Okay, so these right here, these are the major pathways for excretion of histamine. Let's do a quick recap of what we've seen. So I have histamine, right? And histamine can react with histamine N-methyltransferase. That basically puts a methyl group at this position right here. It replaces this hydrogen on this nitrogen of the imidazole ring with a methyl group. It's an S-adenosylmethionine-dependent methyl transfer, just as we would expect, and you get SAH, S-adenosyl homocysteine, as a product. That gets metabolized into the SAM cycle. Okay? Then an alkylamine oxidase, which is oxygen and water dependent, will essentially oxidize the amine on the alpha carbon. Keep in mind, this is the alpha carbon right here. It will oxidize the alpha carbon into a Schiff base, then perform a Schiff base hydrolysis, and we would expect to get aldehydes, and we do get aldehydes. The aldehydes then get oxidized into carboxylates. That's catalyzed by either aldehyde dehydrogenase or xanthine oxidase, surprisingly. Okay? And then when you generate N-methylimidazole acetate, you're done. This right here, this guy is a product that can be excreted, and it will be, by the kidneys. But in the other case, we can go one step further and react it with a phosphoribosyltransferase, which uses PRPP to transfer a ribose 5-phosphate moiety onto the imidazole ring, and we end up generating something called imidazole acetate ribose 5-phosphate. So this is imidazole acetate ribose 5-phosphate. And there's various other ways you could name that, but that's a perfectly reasonable name. So I hope this video gave you a little bit of intuition on histamine catabolism. See you in the next video.